Now, welcome to all of you to today's session. We will dive into the interesting word of enzymes. You see that uh, as we know that the simple life forms generally utilize energy from their surrounding. For example, many of us you consume significant quantity of sucrose, which is found as a common table sugar as a source of energy, typically through the sweetened foods and beverages. So the breakdown of this sucrose into carbon dioxide and water in the presence of oxygen is an energetically favorable process and release usable energy for carrying out various functions, movement, taste perception and vision. However, while a bag of sugar kept on a shelf for years without visibly undergoing this conversion, there is no change at all. So, however, what happens once consumed by these sugars are consumed by human or virtually any other organisms, generally sucrose releases its stored energy within seconds. So we can say that the key difference lies in the catalysis. Without catalytic process such as oxidation of sucrose, these chemical reactions would not proceed at a meaningful rate, and thus making the life unsuitable. So basically all the life forms relies on a series of chemical reactions, and majority of which don't occur spontaneously, and many of these reactions are very slow to support life on their own, and thus require catalyst to significantly speed them up, and these catalysts can be described as the acceleration of a chemical reaction by substance that remain unchanged. And this process enables essential biochemical reactions, which is vital for life. So enzyme basically serve as a catalyst for nearly all the chemical reactions within living organisms. So one thing you must be very clear that these enzymes serve as a sort of catalyst to carry out various chemical reactions in the, you can say, in the human body or in the plant body. So enzymes have been recognized for over more than a century. There's a French chemist, A. Payen, who discovered the enzyme diastase and dates back to in the year 1833. Then L. Pasteur later recognized that enzymes played a crucial role in the fermentation, but initially he believed that their catalytic function was closely linked with the structure and life of yeast cell, and he just revealed this fact in the year 1860. Then moving ahead in 1877, basically a German physiologist, W. Kuhne, coined the term enzyme, derived from the Greek word for leaven, to describe this process. Then German biochemist Edward Buchner demonstrated that cell-free extract of yeast could ferment sugars into alcohol and carbon dioxide, and Buchner basically named this preparation zymase. So this groundbreaking discovery carried out by Buchner was the first indication that enzymes could operate independently of cells. Later in the year 1926, American biochemist J.B. Summer isolated enzyme urease, which is the first enzyme isolated in pure crystalline form from the jack bean. Then J.B. Summer suggested that this molecule was a protein, and contradicted common belief at that time that it was the part of some cells. Then later on, pepsin, chymotrypsin, and then trypsin were also crystallized successfully, confirming their protein composition, and this establishes the protein nature of enzyme. Basically, proteins are the predominant catalyst, and these are also ribonucleic acid known as ribozymes, which can exhibit enzymatic activity and generally, but they are less encountered. So, in this chapter, we will focus on reaction uh, catalyst of biological system called enzymes. Uh, Basically, learning about enzymes is really very useful. Sometimes enzyme deficiency can cause various health problems. On the other hand, you see that having too much of an enzyme can also lead to various health issues, and doctors can measure enzyme levels in our blood or tissue to help to diagnose certain sort of specific illness. Many medicines works by interacting with enzymes, and in addition to this, enzymes are also important for practical tools in chemical engineering, food technology, and agriculture. So first we will talk about the properties of enzyme. Then in the coming lecture, I will discuss the nomenclature and classification and how these enzymes basically work. Then we will dive into the enzyme kinetics, which is basically about how fast enzymes do their job. After that, we will look at some specific examples to understand how enzymes basically work. And then finally, we will discuss in our coming lectures how enzyme activity is controlled. So first, we will begin today's lecture with the properties of enzymes and the principles underlying their catalytic power. Now, before discussing the properties of enzymes, we should learn about some basic terminologies that is you are likely to commonly encounter while doing the course of this lecture, particularly related to enzymes. So basically, an enzyme is once an enzyme is denatured or broken down into its subunit, basically enzyme loses its catalytic function. Therefore, enzymes are basically micromolecular biological catalysts that play a crucial role in facilitating nearly all the chemical reactions necessary for maintaining homeostasis in organisms without themselves being changed in the process. So, a large enzyme molecule basically primarily composed of one or more chains of amino acid connected by peptide bonds, and which relies on its specific amino acid sequence to determine its characteristic structural folding pattern, crucial for enzyme specificity. So, any sort of alterations to the enzyme, such as changes in the temperature, pH, could lead to denaturation, or the protein structure loses its integrity, generally, and enzymatic functions. While denaturation, you see, sometimes could be irreversible, but it is not always the case. Then you see that the active site present in the enzyme is very much essential for its proper activity, and these active sites are a specialized region where generally substrate molecule bind and undergo chemical reactions. Then residues that catalyze the reaction of the substrate serves as a catalytic site. Within the enzyme, the active sites are present, and typically the active site appears as a groove or pocket within the enzyme, which may be is situated in a deep tunnel or between interface of the multimeric enzymes. And in addition to the active site, enzymes also possess an allosteric site where an effector molecule can bind, provide another means of enzymatic regulation. So this allosteric modification is typically observed in proteins with generally multiple subunits. These interactions, known as allosteric interactions, are commonly found in metabolic pathways and serve to regulate one step of a reaction by influencing another step. So, this flexibility allows enzymes to engage in a variety of molecular interactions beyond those occurring at the highly specific active sites. Some enzymes contain an additional chemical non-protein component known as a cofactor. So, try to understand what this cofactor is. So, basically these cofactors which directly participate in catalytic event and is generally essential for proper enzyme activity. Cofactors may be either coenzymes, which are organic molecules such as vitamins, and they are thermostable and loosely bound to protein part, or these cofactors may be inorganic metal ions such as potassium or iron, known as metal activators or simply activators. Some enzymes require both types of cofactors. Cofactors could be either tightly or loosely bound to the enzyme. When they are tightly bound, they are termed as prosthetic group. And these prosthetic group are organic substances that are, as I mentioned, thermostable, firmly attached to the protein or apoenzyme portion. So, the entire basically active complex consisting of the protein portion and the, that is apoenzyme, and the cofactor, coenzyme and prosthetic group or activator is known as haloenzyme. In other words, the combination of apoenzyme and cofactor forms the haloenzyme, or you can say that now zymogen, basically as I mentioned earlier, the zymogen refers to an inactive precursor form of an enzyme, which is typically not always a proteolytic enzyme or proteinase. Some zymogens are named by by appending the suffix ogen to the enzyme's name itself, such as trypsinogen or pepsinogen, while others are denoted by prefix pro, as seen in the pro-collagenase or pro-carboxypeptidase. So, these are the basic terms. You must be very clear about this. What is cofactors, what is activator, what is basically these cofactors, implicit, activator, coenzymes and prosthetic group. Now, I will discuss one by one the various distinctive characteristics of enzymes one by one. First one is catalytic efficiency of the enzymes. So, you see that enzymes show a very remarkable catalytic power even present in small quantities. Therefore, they basically enable the conversion of large amount of substrate into product, and consequently enzyme catalyzed reactions proceed significantly at faster rate with acceleration ranging from 10 to power 3 to 10 to power 8 times faster compared to generally uncatalyzed reactions. So, the turnover number of enzyme represents the number of substrate molecules converted into products per enzyme molecule per second, and typically this turnover number ranges from 10 to power 2 to 10 to power 4 per second. For example, catalase is a very widely studied enzyme, has a turnover number of 4 into 10 to power 7. This is the highest among known enzymes, while lysozyme exhibits the lowest turnover number, and that is about 0.5 into 10 to power 7 per second. So, you must remember which enzyme has highest turnover number, which has least turnover number. Now, what are the active sites of enzyme? Basically, you see in this figure that substrate molecule binds to a peculiar site, and so where substrate can bind question arise, whether throughout the enzyme part or at specific location. So basically these enzyme molecule, you see that the enzyme molecules possess a unique region known as active site, which is a specialized pockets created by folding of the protein structure. Within this active site, basically specific amino acid side chains are situated, which facilitate both binding of substrate and then carries out the catalytic reactions. When the substrate you see that interacts with the enzyme, it forms enzyme substrate complex. This binding event is believed to induce a sort of conformational structural alteration in the enzyme, which enhances the catalytic activity. Basically, the enzyme substrate complex undergoes conversion into an enzyme product complex, which subsequently dissociate, releasing the enzyme and the newly formed product, enzyme and product. So this is the chain of reaction generally involved right from the reaction, enzyme substrate reaction, and then enzyme and product, enzyme are released and product is formed. Now third peculiar feature of enzyme is reversibility. Enzymes basically you see possess a very remarkable property known as reversibility, allowing them to catalyze reaction in both forward and reverse direction. It means that the same enzyme can facilitate both formation and breakdown of molecules depending on the prevailing conditions within the cellular environment. For example, you see that the enzyme lactase breaks down the lactose sugar found in the milk into its component sugar, glucose and galactose. However, under certain conditions, what happens, such as in the absence of lactose, lactase can also catalyze the reverse reaction where glucose and galactose combine to form lactose. Similarly, the enzyme carbonic anhydrase speeds up the conversion of carbon dioxide and water into carbonic acid. However, under certain conditions, different conditions, carbonic anhydrase can also facilitate the breakdown of carbonic acid back into the carbon dioxide and water. So you see that this reversibility is very unique property of enzyme, which is crucial for maintaining biochemical equilibrium within the cells and allows for dynamic regulation of different metabolic pathways. This feature also enables cells to respond to various changing environmental conditions and ensures that biochemical processes could occur efficiently inside the cell. Now specificity of enzymes is a very unique feature. So you see that enzymes also exhibit remarkable specificity, means they selectively recognize and bind to a specific substrate to catalyze specific reactions. This specificity ensures that enzymes only interact with the molecules that they are designed to work on, preventing unnecessary reactions and maintaining the efficiency of biochemical processes. For example, the enzyme lactase specifically targets the sugar lactose and breaks down it into component sugars in glucose and galactose. Similarly, you see in the reaction, lactose is broken down into glucose and galactose by enzyme lactase. Similarly, the enzyme sucrase basically act on sucrose, cleaving it into glucose and fructose molecule, glucose and fructose molecule. So this specificity allows each enzyme to carry out its designated function without interfering with other biochemical pathways. The specificity of enzyme is crucial for the proper functioning of biological systems, which ensures that essential metabolic reactions occur accurately and efficiently, contributing to overall cellular health and function. Now, another feature of enzyme that enzymes rely on molecules other than proteins for their activity. You see that the term holoenzyme refers to the complete active enzyme along with its non-protein component. You see, holoenzyme. This is the whole active enzyme along with complete active enzyme along with non-protein portion. So, opposite to it, an enzyme lacking its non-protein component is termed as apoenzyme. This is apoenzyme, and generally it is in inactive form. When the non-protein component is just I mentioned is like metal like zinc or iron, it is known as activators, and these activators are basically cofactors. On the other hand, if this non-protein part is a small organic molecule, it is called a coenzyme. If the coenzyme remains permanently attached to the enzyme and is returned to its original state after the reaction, it is called prosthetic group, such as FAD. Coenzymes are basically often derived from vitamins, as I mentioned earlier. For example, niacin is the precursor for NAD, while riboflavin is the precursor for FAD. Now, these are some examples of inorganic ion that serve as a cofactor of enzyme, like copper serves as a cofactor for cytochrome oxidase, iron in the proper functioning of cytochrome oxidase, catalase, peroxidase, like potassium is involved for proper catalytic action of pyruvate kinase, like magnesium, manganese, molybdenum, nickel, selenium and zinc. They are specific for the proper catalytic activity of the enzyme. So, you should remember that which activator or cofactor is specific for proper catalytic activity of which enzyme. So, you should, from this table you see the role of different activators. Of course, these activators are cofactors that regulate the proper activity of coenzymes, cofactors. These they are regulate the proper activity of enzymes. Now, these are the differentiating point that exist between cofactors, coenzymes, and in what respect these cofactors and coenzyme differ from each other. And you see from this table it is very much clear. These cofactors could be organic, inorganic molecules, but coenzymes are always organic molecules, and these cofactors they are inorganic and organic cofactors, and coenzymes are actually you should not confuse with the differentiating point here. You see that cofactors include, as I mentioned earlier, metal activator, coenzymes and prosthetic group, but of course they differ from each other. So, cofactor basically implicit activators, coenzymes and prosthetic group. Now, how activity of enzyme could be regulated? So, you see that enzyme activity can be regulated, you can say that its activity could be increased or decreased. So, the rate of product formation responds to cellular needs. Basically, enzymes possess a crucial regulatory property that allows them to adjust their activity level in response to changing conditions within the cell or organisms. This regulation basically ensures that metabolic processes are finally you see tuned to meet the dynamic need of the cell and dynamic need of the cell and maintain the overall cellular homeostasis. There are several mechanisms by which enzymes can be regulated. One is allosteric regulation. You see, allosteric regulation. So, some enzymes have very specific regulatory sites known as allosteric sites. This is distinct from active sites. You should remember this fact. These allosteric sites, they are distinct from the active sites. Regulatory molecules called allosteric modulators bind to these sites, or you can say allosteric sites, and can either enhance or inhibit enzyme activity. This form of regulation allows for rapid response to fluctuations in cellular conditions. Second is feedback inhibition. In many metabolic pathways, the end product of the pathway acts as a feedback inhibitor by binding to an enzyme early in the pathway and thereby reducing the activity of the pathway as a whole. This basically helps to prevent the overproduction of certain molecules and conserves resources within the cells. Now, third kind of enzyme regulation is covalent modification. Enzyme activity could also be altered through covalent modifications such as phosphorylation or dephosphorylation. I have discussed it earlier. Addition or removal of functional group can change the enzyme's shape or charge and then affecting its activity. This basically mechanism is commonly used in signal transduction pathways to regulate cellular responses to different external stimuli. And then gene regulation is also very crucial. You see that the expression of enzymes can be regulated at gene level. Various transcription factors and regulatory proteins control the rate of gene transcription, determining the amount of enzyme produced by the cell. This form of regulation basically allows for long-term adjustment to enzyme levels in response to development, environmental or hormonal cues. So you see that overall regulatory property of enzymes ensures that biochemical pathways operate efficiently and adaptively, allowing cells to maintain equilibrium in the face of changing internal and external conditions. Now, localization of enzymes within the cell is also very crucial. Where is the seat of particular enzyme? Just I discussed different respiratory pathways and cycles and electron transport chain. Their localization is different. So, localization of pathway and cycle is very crucial. So, enzyme localization is also very important. So, numerous enzymes are situated in distinct organelles within the cell, and each organelle in the cell houses specific enzymes that is tailored to its unique environment and function. For example, in the cytosol, many enzymes are found freely floating in the cytoplasm, where they are used for catalyzing specific metabolic reactions. These enzymes they are involved in glycolysis, pentose phosphate pathway, and protein synthesis. Cytoplasmic enzymes are generally involved in central metabolic pathways that generate energy and building blocks for different cellular processes. Now, you see that these are the specific enzymes. Just I mentioned that they are localized in the cytosol, or you can say the seat of localization of these enzymes are cytosol. Now, mitochondria also provide seeds for various enzymes. You see that mitochondria is basically known as powerhouse of the cell and contains enzymes involved in cellular respiration, where generally energy is generated in the form of ATP. For example, enzymes like citrate synthase, succinate dehydrogenase, and ATP synthase are located within the mitochondrial matrix and inner membrane, facilitating the production of ATP through oxidative phosphorylation. We have discussed this aspect in our previous lectures. Now, next organelle is chloroplast. In plant cells, you see that chloroplast contain enzymes essential for photosynthesis, the process by which plants generally convert light energy into chemical energy. Enzymes such as Rubisco, ATP synthase, and various enzymes involved in the Kenville cycle are localized within the chloroplast stroma and thylakoid membranes, where they drive the synthesis of carbohydrates from carbon dioxide and water. Then next cell organelle is endoplasmic reticulum. Endoplasmic reticulum basically houses enzymes involved in protein synthesis, folding and modifications. And ribosomes, you see, that attach to the rough endoplasmic reticulum synthesize proteins, while enzymes within the endoplasmic reticulum in the endoplasmic reticulum lumen assist in the protein folding and post-translational modifications such as glycosylation and disulfide bond formation. Then another cell organelle is Golgi apparatus. Enzymes present in the Golgi apparatus are basically responsible for further processing and sorting of proteins synthesized in the endoplasmic reticulum. Enzymes such as glycosyl transferases and proteases basically modify protein glycosylation pattern and then cleave signal peptides, preparing proteins for secretion, or you can say localization to specific cellular compartments. Then lysosome also houses specific enzymes involved in digestion and recycling. For example, acid hydrolases such as proteases, nucleases and lipases basically degrade macromolecules from endocytosed material, damage organelles or cellular debris, allowing for nutrient salvage and waste disposal. So these lysosomes, you see that possess very crucial enzymes which are involved in cycling of waste material, waste disposal. So you see that this is the spatial organization of enzymes within different cellular compartments, which allows for the compartmentalization of metabolic processes, for efficient utilization of resources and coordination of complex cellular functions, contributing to overall functionality and adaptivity of the cell. So you see that this spatial localization of enzyme plays a very crucial role for compartmentalization of different metabolic processes inside the cell. Now susceptibility of enzyme to heat and inhibitors. You see that inhibitors enzyme exhibit high sensitivity to heat and then making them thermolabile or altered by heat, and heat basically also inactivate at very low temperatures. Generally what happens at high temperature or elevated temperatures, typically around 60 to 70 degrees C, enzymes are commonly denatured and destroyed. Enzymes generally with lower molecular weight tend to be more resistant to heat, and then enzymes are also responsive to various inhibitors. Certain enzymes could be inhibited by certain specific inhibitors, making it to become inactive, such as various toxins can irreversibly destroy enzymes, permanently inhibiting their proper functioning. So how you see that this slide shows how the enzyme activity increases with the increase of temperature up to certain level, then further increase in the enzyme activity, that is roughly around 40 degrees C, further denatures the protein part, thereby rendering it to slowly decrease the activity of enzyme, you see, from 40 degree onward. So enzymes are basically very sensitive to temperature and presence of inhibitors. Now next property of enzyme is colloidal nature. Colloidal nature of enzyme, basically enzymes exhibit colloidal nature, which are substances dispersed in a medium, and enzymes are typically large protein molecule dispersed in an aqueous environment within the cell, and these basically colloidal nature of enzyme allows to maintain their catalytic activity and structural integrity within the cellular environment. And you see that very important characteristic of enzymes are like, one is dispersion, which is of course mediated by colloidal nature. Dispersion, actually enzymes are dispersed in the cytosol within specific organelles of the cell, forming the colloidal suspension. This dispersion property of enzyme ensures that enzymes are evenly distributed throughout the cell, and thereby facilitating efficient catalysis of biochemical reactions. Then another property of enzyme is solubility. Enzymes are soluble in water as they are composed of amino acid that contains polar functional groups. This solubility basically allows enzymes to interact with water molecules and substrates, making, or you can say enabling them to catalyze reactions in the aqueous environment of the cell. Another property is hydration. Enzymes generally have hydrated surface due to the presence of hydrophilic amino acid residues. This hydrated layer basically helps to maintain the solubility of enzymes by preventing aggregation and denaturation, which facilitates interaction with the water molecules and substrate during the catalysis reaction. Now, large surface area. Enzymes generally have large surface area to volume ratio, which increases their accessibility to substrate and enhances their catalytic efficiency. Basically, this large surface area allows enzymes to accommodate multiple substrate and facilitate complex biochemical transformation. So, overall the colloidal nature of the enzyme plays a crucial role in their function within the cell, ensuring optimal enzyme catalysis activity, stability and interaction with the substrate in basically aqueous environment. So, this is all about the properties of enzymes and other aspects of enzyme catalysis, nomenclature, catalysis, all these aspects I will discuss in my coming part of the lecture. Thank you very much.